again, my lovely gardening friends. How are you today? I'm Mark. I'm a biotech geek and your gardening freak. Today we are still talking about this lovely plant, the poinsettia, because many of you, in fact, have contacted me and asked me, hey Mark, can you tell us, can you explain us how on earth are they able to produce those lovely colors with the bracts? These are not flowers, these are bracts. Well, those bracts are so beautiful and they have so many colors. Are they found like that in nature? Do they find some cream poinsettias or white poinsettias or apricot poinsettias or orange or fluorescent red poinsettias in nature? Do they find that in Mexico? And I say, well, no, not at all. And you hit the mother lode with this question, because you are bringing me to talk about how we create new plant varieties. Well, how is it done? Well, to explain that, we just need to go back and review a few little notions of biology. And this notion has to do with selection. Okay, so there are two types of selection. The natural selection, and you have the artificial selection. So let's start with the natural selection. Well, it was discovered by a gentleman. His name was Charles Darwin. Yes, everybody knows or have heard about Charles Darwin. He published a book that describes the origin of species through the process of natural selection. And in his observations, in his writings, Charles Darwin noted that, well, the environment is very difficult sometimes. It is a hostile environment, and it is a harsh environment, and only the fittest can survive. And this explains why you have new species. Well, the modern theory of Darwinism simply tells us that no, it's not the survival of the fittest. It is the survival of the best pre-adapted individuals. Those individuals that have a pre-adaptation or a predisposition to survive to different types of environmental changes. And this occurs during long periods of time. So this is how species can evolve and transform themselves over the ages to adapt to an environment that's always changing. Does it sound familiar? Yes, it does sound familiar, isn't it? Because we're talking a lot these days about climate change, the changing of the climate. Well, it is not a new thing. It has happened before, and it will keep happening further in the future. So that's enough for natural selection. Now, let's talk a little bit about artificial selection. It takes a while, it takes a long time. It took Sir Charles Edward Saunders years and years and years of crosses and mastication and sowing and harvesting these wheats in order to find the right one. Okay? Nowadays, we are not that patient. We really want to have our results as quick as possible. So for that, we have to rely on the technologies of genetic engineering and molecular biology. And for that, in the 1960s, there was a very interesting discovery that was made. There's a bacterium living in soil, okay? It's a soil dweller, and it is able to transform a plant cell, okay? And this bacterium has a strange name. It is Agrobacterium Tumefaciens. Okay, Tumefaciens, what does it mean? Well, Agrobacterium that causes tumors. Yes, the tumors in plants can be caused by bacteria, they can be caused also by fungi, but Agrobacterium has a very special genetic machinery that allows it to take control of a plant cell at a site of wound or injury. So the bacterium just sticks to the cell wall and it makes enzymes in order to dissolve the wall and to dissolve the plasma membrane and then inject its DNA in the cell. This DNA goes in the nucleus and takes control of the cell, which starts to divide and divide. So this kind of phenomenon has been discovered in the 60s, and in the 1970s they've been able to use it in order to make some genetic engineering tools that allow biologists to put genes in the bacterial chromosome so that it will be injected in the plant cells, and there you go, you have a transformed plant cells. And those plant cells are not transformed to become cancerous, okay? They are not transformed to become sick. They are instead transformed in order to express some interesting proteins. Because you see, when you do molecular biology, everything is viewed at the genetic level. So no matter where the genes come from, they can come from jellyfish, they can come from shellfish, they can come from another plant, they can come from a virus or a fungus or a giraffe or an elephant or a lion, whatever origin it is, it can be placed inside of a plant cell with Agrobacterium Tumefaciens. So this is a beautiful piece of genetic engineering, and this answers the question how they are able to create those fantastically beautiful Poinsettia varieties, also petunia varieties or geranium varieties, and even wheat, corn, barley, potato, tomato. This is how they do it. Okay, they do it in a petri dish, and they are very patient at selecting the right mutants. And the beauty of cloning genes inside of plant cells is that from only one transformed cell you can generate the entire organism, with its roots, with its stems, with its leaves, with its buds, with its flowers, and with its tasty fruit. Okay? One cell only. Understandably, this is absolutely impossible to do that with animal cells. For instance, you cannot take cells from the kidney of a rat or the liver of a cow or the brain of a horse and generate an entire animal with it. It's impossible. You can only do that with a fertilized egg. But for plants, any cell will do. So they take leaf parts, they cut them into little pieces, place them in petri dishes, put a solution that contains Agrobacterium, and voila, you have your transformation. And this is how they do it. And the problem with tampering with the genetic material of living things is that you don't know exactly where the pollen of the plants will end up in nature. So they might confer their wonderful capacities to other plants, like for instance the corn plants that are genetically transformed to be resistant to glyphosate. Well, I mean, when the corn makes its flowers, its pollen is spread throughout the biosphere, and finally the broad leaf plants are able to get this pollen, and lo and behold, you see mutants of these plants being able to resist to glyphosate. So this is a big problem, and as far as I'm concerned, I am not very well convinced that the fact that we should really tamper with the genetic pool of living things just for the sake of profit. It is not the way to go. The agriculture of the future will be and is presently being centered around the living soils. Yes, and I'm still talking, and I will keep talking about the living soils forever, because this is the agriculture of the future. So don't bother transforming plants in a laboratory when all we need is found in the very ground we walk on. The soil itself is a universe of beneficial microorganisms, and we can use these microorganisms to increase soil fertility. You cannot imagine the amounts of living organisms, or, well, for the most part, most of these microorganisms don't do much for plants. And we don't even know what they are for. But a few of them are very, very well known to science. And they are found in soils. And with the Bioponic soil conditioner, what you do is you encourage those beneficial microorganisms to do their wonderful functions so that the plant will be fed 7 days a week, 24 hours a day, as long as it is kept alive and well with the Bioponics soil conditioner. So it is a very, very important aspect, because you see, what we see in the broadacre agriculture methods is that they are not adapted to what is happening presently with the climate change and the mechanization of agriculture. The soils are not able to withstand those types of practices. Okay, the soil finally becomes like a support on which you spread some chemical fertilizers. At one point you will kill the life forms that are found in the soil. So we have to prevent this for the future generations, and for that we have to bring back the life that is found in the soil. So it is a very important aspect for food resiliency, food sovereignty, urban agriculture, broadacre agriculture, as well as organic gardening. So a soil conditioner like the Bioponics soil conditioner will keep your plants healthy, will keep your soil healthy, no matter if it's contained in a small container or a larger container like a flower box, or even in the ground itself. So keep your soil alive and give your plants a favor with the Bioponics soil conditioner. It is inexpensive, it is extremely efficient, and it is safe for humans, for animals, for children, and for pollinating insects. Let's not forget about the pollinating insects, because the poinsettia has developed those lovely bracts to attract the pollinating insects. And this is what explains the tremendous success of flowering plants in nature. 9 out of 10 plants are flower bearing. So this is it. Thanks for watching, and also thank you very much for sharing this video. It will help a good cause, the cause of food sovereignty, organic agriculture, and urban agriculture for all, because it is the green thumb for just a few greenbacks. And if you don't believe me, well, I've got a bridge in Brooklyn to talk to you about, because it is a sure thing. It is tried, tested, and true, and it will stand the test of time. So thanks for watching, and please share those videos, and we will see you soon. Goodbye now.